Okay, so now we're going to pick up with chapter 11 and talk about the microbiome, and then also talk a little bit about the autism work that I had mentioned at the beginning of the semester. And I said if we didn't get to it, then I would talk about it when we talked about the microbiome, and here we are. So today in the section 11.1, we're going to differentiate amongst the terms colonization, infection, and disease. Enumerate the sites where normal biota is found in humans, and discuss how the Human Microbiome Project is changing our understanding of our relationship with bacteria, of our normal biota. Okay, so humans and other mammals have the type of physiology that they have because they have been in intimate contact with microbes for millennia, and in particular their own microbiome from pretty much the moment of birth. So the human microbiome, the term, means the sum of all of the microbes found on and in a normal human being. And what we've discovered after we started looking at the relationship between the microbiome and the human is that these are critically important to the health and the functioning of the host organism. As an example, I told you that I live on a farm, and baby goats, if they do not get their mom's colostrum within the first 6 hours of life, they will die. They never recover because their microbiome is not properly set up, so they're not able to digest their food. So in a lot of organisms, this is an incredibly crucial relationship, and the formation of the microbiome is really, really important. Okay, so for the most part, our resident microbiota colonize us for the long term and do not only do not cause disease, but in many cases really benefit us. The E. coli in our gut, for example, produces most of the vitamin K that we have in our body. So these are good guys. These are, symbiotes. These are not even just, even though they're often referred to as commensal bacteria, they're not. They have a really important positive relationship with us. Um, so in order for infection to occur in these places, the microbes not only have to get past the host defense and enter the tissues and multiply, but they also have to get past the microbiota. So we talked about a lot of the microbiome, a lot of the bacteria that live long-term in niches, being in stationary phase and basically having colonized a particular area. Well, again, when bacteria go into stationary phase, they start to produce things like antibiotics. And also just physically being there, they own the real estate. There is no way that a pathogen is going to be able to attach to skin that is covered by this microbiome. Okay, so disease is a term that is a deviation from health. It's a pathologic state that results when the cumulative effects of an infection damage or disrupt tissues and organs, okay? And there's a difference. Every infection does not necessarily lead to disease. I hate to think of the microbiome as being an infection, but by and large it is. This is tissue that is completely colonized by bacteria. And not only does it not cause disease, but it actually is health promoting in many cases. An infectious disease is a pathogenic state caused directly by microorganisms or their products. And infectious being something that can be also transmitted from one organism to another. Okay, so the Human Microbiome Project was started about 20 years ago, and it's been a worldwide effort, and it has looked at all aspects of the microbiome. So basically, the first step was just to characterize microbes living on and in human bodies when they're healthy. So what should our microbiome look like? And I will tell you that there's huge, huge variation between people. So if you think about it, the outside of our tissues are covered with various different sugars and other protein products that are based on our own genetic code. If you think about how bacteria attach to our tissues, it's all based on the things that are on the outside of cells. Okay, so there's a huge difference between people, based on their blood type, based on their um, histocompatibility complexes that are a normal part of the outside coating of our cells. And so they will have a preference for different microbes to be part of their microbiome. So the next question was, once you know what the microbiome perhaps should look like, how does that microbiome differ with various diseases? So again, you have a lot of difference just between people, but then you have the disease versus healthy state. Um, and these techniques required some of those really big genome sequencing uh, techniques that we just finished talking about. Large sequencing information. You needed to have a really good human genome map, a human genome map that that had to be increased. So initially we had a map of one person, and then we maybe added a couple more. Well, what is the difference between people of different races, between people of different um, uh, geographic origins, people who eat different foods, all of these things play into the components of the microbiome. And so all of those questions had to be answered, and all of the differences that are caused by non-genetic things had to be separated out as well. So we use those big data tools that are necessary when you have these enormous amounts of data, as with a lot of these large sequencing projects. So, what are a couple of things that we learned based on the Human Microbiome project? So, we already knew from when we put together the first human genome that there were surprisingly few genes on the human genome. There were about 21,000 protein coding genes. However, if you take a look at all of the bacteria in and on our body, there are 8 million protein coding genes with the microbiome. So all of the bacteria that live in and on us dwarf the number of proteins that are produced by our own genome. We also learned that microbes are found in places that were previously thought to be sterile. So we always considered the bladder to be sterile. We always considered the lungs to be sterile. And what we discovered is these places are not sterile. They have a microbiome that differs between healthy and diseased individuals. We also found that there are a lot more viruses that don't seem to have a huge impact on us, but they're present. So there are about 100 million viruses per gram of human feces. And we found that a lot of healthy people seem to harbor potentially pathogenic bacteria and other dangerous pathogens in fairly low numbers. So it's entirely possible that you have MRSA on your skin. It's possible that you have C. diff living in your gut. But because of the way the microbiome protects our tissues, is literally they take over the real estate, they protect us from the pathogens which might otherwise attach and invade and cause disease. And we learned that the makeup of one's intestinal biota can influence overall health. Okay, so we basically already knew that our skin was totally covered with bacteria. We knew that a lot of the outer surfaces of our body, so the upper respiratory tract, so we knew that in our throat and in our nose and our gastrointestinal tract, we knew, so we know that teeth are completely covered with bacteria. We knew that the colon had a lot of bacteria. We knew that external genitalia, we knew that the vagina was covered with bacteria. And in fact, this is one of the ways that babies first get their microbiome, is when they pass through the birth canal as they're being born, and they swallow the bacteria that's around there, and that's what's the initial colonization of their gastrointestinal tract to help them digest their food. The ear canal we knew. We also knew that all of the wet surfaces around the eye had bacteria. But again, based on actually swabbing and checking, we've discovered that there are other places that have normal microbiota, or at least the DNA from the microbiota, places that we thought were sterile. So lungs, bladder, breast and breast milk, amniotic fluid. So we had always considered amniotic fluid to be sterile, but nope, that is one of the sources that the fetus gets their microbiome established. Um, okay, and we have also found DNA from microbes, not necessarily the microbes themselves, but we found products from the microbes in the bloodstream and in the brain. And I will also add that I personally read a research study that talked about some bacterial metabolites that were processed by the liver. And we found these metabolites in the bloodstream, and we found the processed metabolites not only, coming out of the liver, but we found it in all of the excretory. So we found it in urine, and we found it in feces that have been processed by the liver and removed from the body as part of that processing. Okay, so the human body has a variety of environmental niches, and these niches vary from place to place. So the inside of the ear canal looks very different than the inside of the colon. So these places vary in temperature, in pH, they have different kinds of nutrients, they have different oxygen levels, and so therefore they support a wide range of microbes depending on the nutritional requirements of the microbes, whether or not they like oxygen, all that sort of thing. So different places on and in the body attract different kinds of microbes. And as I mentioned, we have microbial antagonism. So the good guys in the ideal situation have taken over the real estate, and they don't allow the bad guys to attach. So this is actually a part of our immune system, is the ability to fight off these pathogens by just simply not allowing them any place to attach. So the normal biota have formed these colonies, and in some cases they've formed biofilms, and they've done all of these things to take over that section of whatever tissue that they are part of. And because of that, they're unlikely to be displaced by incoming microbes. There's just no place for those microbes to attach. Um, and also the chemical or physiological environment, which is created by the microbes, is hostile to other microbes. So in some cases, they produce a slightly acid environment. Well, unless another microbe is able to live in an acid environment, that is enough to keep them from being able to infect. So most normal biota is beneficial to us. Worst case, it's commensal. It's just there, doesn't do anything for us, doesn't do anything to harm us. It's just there. And like I say, in many cases, all it's doing is protecting a tissue from being colonized by a pathogen. That's a pretty important thing. And that's true of a host that's basically in good health with a properly functioning immune system. Okay, now what makes us susceptible to infection? So, old age and extreme youth are two things, because in the case of old age, our immune system has diminished. And in the case of extreme youth, it hasn't fired up yet. It has not been exposed to enough pathogens to be able to distinguish what is a pathogen and what is healthy tissue. It just basically, there isn't a whole lot there other than our innate immune system, which we're born with. And we'll talk about that when we get into the next few chapters. There are also cases where you have genetic defects in immunity. So, for example, children born with Down syndrome are often have a reduced immune system. That's just a part of the package that goes along with that. People who have AIDS obviously have a reduced immune system. That's what the AIDS virus does, or the HIV virus. People who have just undergone surgery. Uh, that's a huge trauma to the body. The immune system often is just simply dealing with whatever has occurred because of the surgery. Organ transplants, people are given drugs to reduce the immune system, because the immune system, if it's fully functional, is going to go after that new organ. And so they're given immune suppressing drugs to be able to deal with, to be able to maintain the organ. People with underlying disease like cancer, like a liver that's malfunctioning, diabetes. So bacteria love sugar, and if you have sugar in your bloodstream, oh boy, and if you have sugar in your urine, oh boy, there you go. You have a wonderful environment to form, groups of pathogens that shouldn't be there and would, under normal circumstances, not have the food necessary to be able to establish an infection. Mental stress, so cortisol does a number on your immune system. So pregnancy, in fact, during the second trimester of pregnancy, there is an intentional reduction in the mom's immune system so that the immune system does not attack the baby, which is, a foreign object. The DNA is different. The coding on the baby cells are probably different than the mom's. And if the mom had a fully functional immune system, it would likely attack the baby. Okay, so again, until very recently, the uterus and its contents were thought to be sterile during pregnancy, at least within the amniotic sac. But they did an analysis of newborn stool before their first meal, so they haven't had any input of bacteria, and it shows all kinds of bacteria are in there. So the colonization of the fetus occurs during gestation. And so this indicates that their intestines were colonized in utero, and the bacteria had to get in there. So this is not as sterile an environment as we thought it was. Okay, as I mentioned, that one of the really important steps in creating the microbiome for a newborn is the trip through the vagina. So there's a lactobacillus that provides the baby with necessary enzymes to digest milk. So lacto gives an indication of its preferred food, which is lactose, which is the milk sugar that comes in the mother's milk. Other species protect the baby from skin disorders. Okay, so again, it takes over the real estate on the baby's skin and gives a protective coating. And of course, one of the things that is a downside to having a cesarean section, aside from the fact that you've now had abdominal surgery and all of the things that goes along with that, is that the baby does not get the proper inoculation of correct microbiota, which it would normally get during a trip through the vagina. Now, human breast milk contains about 600 species of bacteria and sugars that the baby can't necessarily digest, but if it has the healthy gut bacteria, it's able to digest it because the bacteria pre-digest some of it. It's able to be absorbed and then able to be digested by the newborn. Okay, so again, this is just kind of a recap of some of the things that we said. So again, we originally thought that the amniotic sac was sterile. That's not the case. During the birth, this is another place that they pick up additional bacteria for their microbiome. Breast milk and suckling on the breast. Um, People who give babies kisses and touch them and all that sort of thing, all of their microbiome is being shared with the baby. And just in the environment, once babies start crawling, man, everything goes in the mouth and they pick up all kinds of stuff from the environment. And, you know, you guys, most of you are not moms and you don't learn that bacteria is not a bad thing. So most first moms boil everything and sterilize everything, and after you've had your second kid, it's like, if there isn't any visible dirt on it, we just take the pacifier and stick it back in the baby's mouth, because this is okay. If the doggy licks them, it's okay. This is a normal part of building a microbiome and building an immune system. Okay, so this is our concept check. Which of the following body sites were previously thought to be microbe-free but harbor at least some normal microbiota? So skin and mucous membranes, external genitalia, the gastrointestinal tract, breast milk, and the upper respiratory tract. And by that we mean the mouth and the nose. So breast milk was something that we originally thought was sterile, at least inside the body, and we have learned that no, breast milk contains quite a bit of a microbiome, and this is a beneficial microbiome for the newborn baby. Okay. So now I want to share with you some of the autism information that I would like to talk about. So this is actually an area of personal interest of mine, and when I originally went to graduate school, this was what I hoped to study. So let me kind of introduce the whole subject of autism. So autism we don't have a particular positive diagnosis. We can't point to a gene. We can't necessarily point to, some kind of an infection or anything like that. It's kind of a diagnosis of exclusion when you've sort of excluded everything else that it could be, and you see these kinds of behaviors, it's diagnosed as autism. So it's characterized by a lot of atypical behaviors. So you have impairment in verbal and nonverbal communication, failure to develop social skills, to be interactive with other people. Often it comes with repetitive gestures or speech. So it's very typical to see a child with autism doing what we refer to as self-stimulating behavior. This is something that stimulates certain nerves, produces certain endorphins, and that's what the child is doing with those behaviors. And typically they're unable to engage in any kind of symbolic or imaginative play. So for example, if you give a young child a little toy car, usually they're going to, run it up your leg and vroom, vroom, vroom. They're going to do something that involves, sort of pretend driving. Whereas if you give a child with autism some cars, they're going to line them up. And so this is not anything related to how cars are used in the real world. So toy cars are a symbol for something that's out in the real world. And this is not anything that they do that. They would do the same thing with, uh, any kind of, well, I guess we don't use VHS tapes anymore, but a stack of DVDs, they're liable to stack them up or line them up. They're not necessarily going to take them and put them into a DVD player. So here is a picture of two siblings, and you can see the young man in the front is looking right at you. He is engaged in a nonverbal communication, whereas his brother in the back is in his own world. This is very typical that they're chewing on clothing, chewing on body parts, whatever. These are very typical kinds of behaviors. Typically, the onset is prior to age of 3, and there is a 4-to-1 bias affecting males. So boy children are way overrepresented in the world of autism. So one of the things that is interesting from a scientific perspective is that gut dysbiosis. And what I mean by that is a gut microbiome that has too many of the wrong kinds of bacteria, so it is imbalanced. Um, so let me introduce one more term called regressive autism. And this describes a child who has grown completely normally up through about 18 months of age. They're saying mama, they're interacting with their parents and their siblings and the family dog and all that sort of thing. And all of a sudden, they lose all of that interaction. And this particular type of autism is often accompanied by very significant GI symptoms. So one of the behaviors that is very typical for children like this is you'll find them leaning over the couch. So what they're doing is they're putting pressure on their gut because it hurts. And, if you have a wound, the first thing you want to do is, put pressure on it, not only to stop the bleeding, but it just makes it feel better. Well, the same thing is true with these children leaning over the couch, and they'll stay there for hours because it makes them feel better. So, 70 to 80% of children with this type of regressive autism show these gastrointestinal symptoms. It is frequently associated with mucosal inflammation. In fact, one gastroenterologist was looking at was putting endoscopes into these kids who were showing these symptoms of gastrointestinal discomfort and disease. And he was finding very specific inflammation at a very particular place in the gut. And he nicknamed it autistic enterocolitis because it was so um, connected with children with this regressive autism. And kids with this regressive form of autism often have immune dysregulation. They have recurrent mucosal infections, and they also show signs of microbial dysbiosis. And again, dysbiosis means you have the wrong complement of bacteria in whatever place it is, in this case, the gut. Okay. So let me talk a little bit about the genus Clostridium. So I talked about it a little bit before, but now I think you're a little bit more ready to understand some of the terms that I'm going to be throwing at you. So Clostridia, the genus Clostridium are they're obligate anaerobes. They really don't like oxygen at all. They're gram positive and they're spore formers. Okay. There are 120 known species, and 35 of them are known pathogens. And these are really serious pathogens in many cases. So Clostridium perfringens is gas gangrene, okay, and necrotic enteritis. So it kills tissue. No, C. diff is how this is typically referred to, because nobody is quite sure if you're supposed to pronounce it in the Latin method, which is difficile, or if you're supposed to pronounce it in the French, which would be difficile. So we call it C. diff, and then we don't have to worry about how to pronounce it. So I'm a Latin person, so Clostridium difficile causes massive, horrible diarrhea and something called pseudomembranous colitis. And what that means is that the gut lining actually separates and forms this separate tube. It's incredibly painful. And this is one of those diseases which is often caught in the hospital. This is a nosocomial infection. And it's passed around within the hospital situations. And so this is a really nasty one, and it's incredibly hard to kill, and it is so painful and difficult to deal with. And of course, Clostridium tetani is our friend tetanus, and Clostridium botulinum is our friend botulism. And these produce very, very powerful neurotoxins. Um, so many species of Clostridium have both an enterotoxin, which means it attacks the gut lining, and a neurotoxin. Okay, these are nasty, nasty bugs. They're incredibly metabolically active, and they produce a lot of toxic byproducts. Okay, so here are some examples of some of the types of chemicals produced by various species of Clostridia. So indoles, which are precursors to neurotransmitters and hallucinogens. So I don't know how many of you are familiar with a drug called LSD. It was really popular back in the 1960s. Never tried it, never want to try it. Um, but basically, a lot of the magic mushrooms that are popular on the street have indoles as the hallucinogens that are part of it. Um, Phenol. Okay, some of you are probably familiar with this product, Lysol. Okay, that's phenol. It will kill off lots of other bacteria. Acetone. You guys are familiar with this product. It's nail polish remover. Okay. And then the last one that I'm going to talk about is ethanol. Okay. So there's a nice bottle of vodka. So I want you to think about somebody who has too many of a bacteria that kicks out Lysol, nail polish remover, and vodka, and you can see how these bacteria could potentially have a really significant um, uh, effect on the nervous system and on the brain and on thinking. Okay. So there is a scientist named Sidney Feingold who has done a lot of work with children with various kinds of disabilities, and in particular looking at the gut. And what he did, now this study was done back in the day when things were being cultured. So he was not doing this particular thing in the age of, high, lots of sequencing, high throughput sequencing. And so he was actually culturing these clostridial species. Okay, and so what he did was he took fecal samples from children with this regressive type of autism and their neurotypical peers, and he cultured them and counted the different species of Clostridia. And just in general, the count of Clostridia in children with autism was just about 10 times higher. Okay, so there was a huge imbalance in just this one genus. Okay, he found 9 species in the kids with autism that were not in the control group at all. And he found 3 species that were in the control group that were not present in the children with autism. And he used a PCR, which we just talked about in our last lecture. But in this case, he did a quantitative PCR. So he was actually counting the copies of various microbes using these probes. So he would find a gene that identified a particular microbe, a particular microbial species, and then he would count those. Okay, and these Clostridia come in clusters of different pathogenic levels. Okay, so cluster one has all the nasty ones. Not all of them, I guess it has quite a few of the nasty ones. He found 9 times higher things like get the one that causes gangrene, the one that causes tetanus, the one that causes botulism, and histolyticum ought to give you a clue that what it's doing is it's going into tissues and breaking open the cells. So it's very damaging to skin type of tissues. Cluster 11 includes C. diff, and that was 3.5 times higher. And there was one that was only found in the kids with autism, and it was 46, or I shouldn't say only, it was 46 times higher in the kids with autism than in the normal controls. And so he actually looked at that particular one as almost a biomarker. If you have really high levels of that, that seems to be associated with autism. And so, this was incredibly interesting research, and the only person who was looking at these things back in the day when he published this back in the early 2000s. Okay, so that is the end of the material for exam 3. Reminder that we're going to be having a study session on Monday and a study session on Wednesday. So please come with all of your questions prepared. I'm not planning to give any lectures on either Monday or Wednesday. Hopefully we'll get a chance to play some games, either Jeopardy or some kind of a fun game to make studying maybe a little bit more fun. But I'm not going to be lecturing. Friday, of course, is going to be your exam. Okay, I will see you guys on Monday. Bye-bye.